See DNA profiling fingerprinting. DNA is the master molecule of all life forms. It constitutes the blueprints of every living organism through which characters are passed generation to generations. However, every living organism differs only due to nucleotide sequences of chromosome. The nucleotides form codes, therefore, this coded genetic information can be profiled to produce most authentic identity of any organism. The complicated technology that facilitates the identification of individuals at genetic level is known as DNA fingerprinting, or more specifically DNA profiling. This genetic analysis is based on identifying tiny segments of the hereditary material which testify the unique molecular signature which cannot be altered. With the help of DNA profiling technology, identity of burnt or unidentifiable dead body can be done. The basic requirements for DNA profiling is the availability of biological samples, which can be anything including blood stains, a piece of hair with its root, few drops of semen, skin cells, a mouth swab, cells of bone marrow of any body tissue. In 1984, for the first time, a British geneticist, Alec Jeffreys, working at the University of Leicester, developed the technique of DNA fingerprinting. This method has been patented, and being used in Europe and America. Its success is based on identification of small segment of DNA which testifies the unique molecular signature that every person on Earth possesses and which cannot be changed in one's lifetime. VNTRs. It is important to note that 99% of base sequences is the same in the DNA of all human beings. Only a short stretches of sequences, which is about 3 billion base pairs, differ person to person. Out of the total DNA, about 1 in 1000 base pairs is a site of variation in the population. This is an unusual sequence of bases of 10 to 15 base pairs long, which is repeated several times. These bases were first isolated and identified by A. Wyman and R. White in 1980 at the University of Utah as hypervariable segments of DNA and termed as variable number of tandem repeats (VNTRs). On the basis of number of repeats present in VNTRs, the length of segment is measured. In different person, the length of VNTR differs, and therefore VNTRs is the key of DNA fingerprinting. It was Jeffreys who first used VNTRs in forensic science. The VNTRs act as genetic markers. The scheme developed by Jeffreys for measuring the length of fragments of DNA containing the repeated sequences is also known as restriction fragment length polymorphism (RFLP) testing. 1. Method of DNA fingerprinting. In this technique, DNA is isolated from blood stains, semen stains, or hair roots of disputed children or any suspected person, and the same from parents, close associates, or relatives of suspected criminals based on the cases. Since hairs contain less amount of DNA, it can be produced in a large amount by using polymerase chain reaction (PCR). RBCs do not contain DNA, therefore WBCs are the source of DNA. Thus the DNA isolated is cut with restriction enzyme and subjected to southern blotting. The DNA bands appearing on membrane are hybridized with 32 probe, washed in water to remove unhybridized DNA, and passed through X-ray. The hybridized complementary DNA sequences develop images prints. Identical prints that contain specific DNA sequences appearing on two X-ray films are identified, and thus identity is confirmed. Probability of two persons having similar sets of base pairs in the same sequence of VNTR of DNA is 1 in 30 to 300 million individuals. In India, facilities of DNA fingerprinting at international level are available at the Center for DNA Fingerprinting and Diagnostics (CDFD), Hyderabad, the then Center for Cellular and Molecular Biology (CCMB), Hyderabad. At this center, the Jeffreys technique is not followed. Dr. Lalji Singh has developed an indigenous technique where BKM DNA probe is used for hybridization of DNA sequences. While he was working on sex determination in snakes in his PhD program in BHU, Varanasi, he found a contrast result. Unlike human and other organisms, the female snake contains XY and the male Y sex chromosomes. A segment of DNA was isolated from sex-determining Y chromosome of female banded krait Bungarus fasciatus, an Indian poisonous snake. This unique segment was named as banded krait minor satellite (BKM). It is similar to sex-determining chromosome in humans. The probe which is used for this purpose is, therefore, called BKM DNA probe. 2. Application of DNA profiling, DNA fingerprinting technology. DNA profiling technology has applications in many more areas as given in Figure 7.10 and discussed below. A. Setting up of genetic databank, realizing the potential of DNA profiling genetic databank is being set up throughout the world. Databases of the genetic fingerprints of criminals have been set up in many parts of the USA and Britain. Forensic Science Services Birmingham, Britain has set up the world's first national DNA database where about 5 million records would be available. It has collected samples of tissues and DNA of suspected individuals has been profiled. The Armed Forces Institute of Pathology, Gaithersburg, USA, has collected DNA samples from every member of the armed services. The samples are stored in freezers in DNA repositories. Each sample consists of blood blots on cards, sealed in individual envelopes. A computer database of all samples has been prepared. B. Reuniting the lost children. DNA profiling technology helped in reuniting the lost children with their respective parents or vice versa who were separated during war, violence, or natural disasters. For example, in Argentina, many people were abducted during military rule 1976-1983. They were presumed to be died. But an Argentinian human rights group, the Grandmothers of Plaza de Mayo, was of the opinion that the children are still alive and adopted by military personnel after killing their natural parents. In 1987, a genetic databank storing blood samples of grandparents and relatives of lost children was set up. By matching the DNA profile of one or more grandparents with that of lost children, about 40 missing children have been reunited with their families. C. Solving disputed problem of parentage, identity of criminals, rapists, etc. To carry out the test, a small portion of blotted blood is punched out from which blood cells are isolated and information is taken from computer databank. The first case from preserved sample was done to identify a soldier who was burnt to death in a car accident. In India, a disputed parentage has been solved. In June 1988, a 4-year female baby named Lakshmi was stolen from Chennai by a missionary and renamed as Mary by the so-called parents. After fur and taking police help, the matter was not solved. Therefore, the real parents went to High Court. The High Court sought help from CCMB. The scientists performed DNA fingerprinting tests of baby and parents, real and false, confirmed the identity of the baby, and submitted the report to the court. On the basis of report of DNA fingerprinting, decision was given by the court, and Lakshmi was handed over to her real parents. This is the first case in India where a court has given decision based on the report of DNA fingerprinting. The second DNA fingerprinting based decision of identity confirmation of a brutally killed lady was done in Delhi in 1995. During those days, the name of unidentifiable dead body was in light. The lady was killed in her home by unknown murderers. The dead body was dumped in a car and taken to a restaurant where it was consigned to the hot flames in a tandoor. Limbs had been brutally severed to unravel the identity. After this incident, no body came F0 to receive the dead body, neither parents nor relatives. Thanks to DNA fingerprinting technology of CDFD that confirmed the identity of brutally killed and roasted lady as SMT Naina Sahni. Her husband has been sentenced to life imprisonment by the Honorable Supreme Court of India. The immigrant dispute, more or less, in every country, there is the problem of immigration. Ultimately, the identity is not confirmed whether they have crossed the borders or are the resident of that country. Therefore, the problem of immigrants can be solved through DNA databank. The animal and plant improvements. 1. Transgenic farm animals. Production of transgenic animals through microinjection techniques will play a significant role in veterinary sciences as well as for human welfare. A farm animal can produce milk containing tons of gram of protein per liter, many times more than a liter of bacterial culture. The possibility of producing human pharmaceutical demonstrated when lactating mice secreted the active tissue plasminogen activator TPA in their milk. The TPA is an enzyme which dissolves the clots responsible for coronary artery blockage that results in heart failure. John M.C. Fusson at Genzyme Company, in collaboration with K. Albert and colleagues of Tufts University, produced the first transgenic goat by using a goat beta-casein promoter gene linked to TPA gene. The transgenic goat produced TPA in quantities as compared to cell culture-derived material. But goat peptide is slightly different from the culture-derived TPA. The goat peptide has 2 chains of peptide, whereas a single chain peptide was found in culture-derived TPA. Therefore, much work is needed in future. Recently, Pharmaceutical Proteins Limited has made contract with Bayer Co. Germany, to produce alpha-I antitrypsin by using transgenic sheep. Alpha-I antitrypsin, a glycoprotein, has now been approved in the USA as a replacement therapy. The scientists are of the opinion that about 12 kilograms sheep lactation alpha-I antitrypsin can be produced. Production of transgenic cow has also been demonstrated in the Netherlands but unsuccessful. The eggs of cow collected from ovaries from slaughterhouses were fertilized in vitro and microinjected with transgenes containing lactiferous gene. The embryos were implanted in the uterus of surrogate mothers. Out of 102 injected embryos, 21 animals were pregnant and 10 calves were born. 2 of these, one male and the other female, carried transgene. The male showed the presence of lactoferrin gene but not the female. A male never gives out milk. Till the animal secretes milk, the experiment is incomplete. But this experiment has given a hope for future. Caseins are the major proteins of the milk. By transferring genetically manipulated casein genes to farm animals, the texture of cheese and heat-stable dairy products can be improved. Casein genes have already been cloned and introduced into bovine genome. Recently, T.J. Pandian and colleagues of Madurai Kamraj University have succeeded in producing transgenic fishes by microinjecting growth hormone gene, human or bovine. A fish produces a large number of eggs at a time. It is much easier to work on eggs for gene manipulation. In developing countries like India, where majority of people suffer from malnutrition, the aquaculture programs can be intensified for production on a large scale of good quality fishes. For details of animal biotechnology and transgenic animals, see chapters 10 and 11. 2. Crop improvements. Population of the country is rapidly increasing day by day. However, the most urgent need to the nation is to meet the food demand of the people. To remain self-dependent, and to make them available with quality food, the biotechnologists have to boost up the gene revolution programs for crop improvement so that the improved crops should have high yield, high amount of digestible and quality proteins, and vitamins, disease pest resistance and drought, herbicide, salt tolerance, etc. These can be done through the transfer of beneficial genes of a prokaryotic microorganism, eukaryotic incompatible plant in a given crop plant, alteration in metabolic pathways and making the plants resistant to invasion of pathogens, pests and herbicides, salt stress. Methods of gene transfer in a desired eukaryotic cell are through agmbacterium, through virus, co-cultivation of cells, leaf disc transformation, electroporation, etc. These are discussed in the following subsections. A. Transgenic plants, see Chapter 13. B. NIF gene transfer, see Chapter 13. C. Phaseolin gene transfer, see Chapter 6, see Sunbean plant. D. Conversion of C3 plants to C4 plants, see Chapter 13. A. Herbicide-resistant plants, see Chapter 13. F. Insect, pest-resistant plants, see Chapter 21. G. Plant improvement through genetic transformation, see Chapter 6. 3. Crop protection. See chapter number 21. A. Abatement of pollution through genetically engineered microorganisms. For detoxification and degradation of toxic chemicals, enzymes are encoded by genes present on plasmids. A. M. Chakrabarti and co-workers in 1977 succeeded in microbial culture which could utilize a number of organic chemicals, toxic in nature, G. The salicylate, 2,4-D, 3-chlorobenzenes, ethylene, biphenyls, 1,2,4-trimethylbenzene, 2,4-S. As rofenoxyacetic acid, etc. Genes responsible for degradation of environmental pollutants, for example toluene, benzene acids, and other halogenated pesticide and toxic wastes have been identified. For compound, one separate plasmid is required. It is not like that one plasmid can degrade toxic compounds of different groups. The plasmids are grouped into 4 categories. 1. OCT plasmid which degrades octane, hexane and decane. 2. XYL plasmid which degrades xylene and toluenes. 3. CAM plasmid that decompose camphor, and 4. Na plasmid which degrades naphthalene. Anand Mohan Chakrabarti, an Indian-born American scientist, produced a new product of genetic engineering called a superbug oil-eating bug by introducing plasmids from different strains into a single cell of P. putida. This superbug is such that can degrade all the 4 of substrates for which 4 separate plasmids were required. Figure 7.11. See Figure 7.11. Creation of a superbug diagrammatic. The plasmids of P. putida degrading various chemical compounds are told for toluene xylene RaSO for 3, 5 xylene pack 25 for 3 1 chlorobenzoate PKF 439 for salicylate toluenes. Plasmid WWO of P. putida is one member of a set of plasmids now termed as toll. Plasmid WWO is propagated in E. coli. For a detailed discussion, see Chapter 25. End of chapter number 7. Problem statements.